somewhat dynamic. So climate change is primarily going to affect aspects of the fundamental nature, things like how warm it is, how much it's raining in a given area. So we want to know, we want to define the fundamental nature of an organism and see how that will change with time. But the correlative model, because using point locations, is by definition using the realized niche. And then you're trying to correlate that with aspects of the fundamental niche and try to decide what happens in the future. And there's a lot of reasons why you won't get what you predict might not really happen. You're just not taking enough into account, and therefore it really raises questions about whether or not those models are useful. However, there are other modeling strategies and other things that can be done. And one of the most highly advocated modeling strategies is to go from a correlative framework to a mechanistic framework. And what this does is it allows you, you have to go out and do experiments, and then you experimentally and via field work actually define the sensitivities of your organisms, figuring out what their physiology will allow, and therefore actually defining the fundamental niche, and you can model how that would then change with climate change. This hasn't been done a lot. One of the best examples is on this guy, Eastern fence lizard. This was work done by Lauren Buckley's lab. And I've got a bunch of maps on the screen, but really all I want to point out is that correlative models over here, mechanistic models over here, they're all for the same species. It's got a huge range on this eastern US, this black line. And the mechanistic models, I'm saying that these are going to work better, but it still depends on actually picking a good model and picking to model the correct aspects of your organism's biology. These are 3 different models that are all looking at different aspects of the organism's biology, and they get drastically different results, or they yield drastically different results. So these models are challenging. They require a lot of extensive knowledge about your organism, things like what processes are actually most sensitive or limiting, what environmental parameters have the greatest effect. You can have things like, it might be mean temperature, it might be the extremes in temperature, it might not be temperature at all. It could be water conditions in an area, precipitation. It also might just be both of those together, but only during a single month of the year, during a certain life cycle. And these traits might not be fixed in an organism. There's a lot of potential for traits to shift within an individual, at least their sensitivities. That's known as phenotypic plasticity. All these things need taken into account, and we really have very little idea, very little consensus of how to do this for any group of organisms. We don't know what traits are likely to be most limiting. We therefore don't really know how to proceed in making mechanistic models that are functional and useful. So that's a large part of my work, is I'm trying to understand what sets the physiological limits on organisms. And I'm going to talk about 3 different aspects of my work that's trying to address this. These are going to be 3 sort of vignettes addressing 3 different questions. First, I'm going to look at whether or not life history stages might have different environmental sensitivities. Generally, mechanistic models that have been done in the past always look at the adults. However, if your adult life history stage isn't the most sensitive, then your model predictions are basically bunk. You're not going to have any useful predictions out of those. Then I want to look at whether or not extreme temperatures are important. We know mean temperatures are likely to rise with climate change, but there might also be some really major increases in extreme events, things like heat waves and droughts and floods and just storms, things like the hurricanes we've been seeing in the news. And then, slightly different, I'm going to look at how behavioral changes might allow these organisms to be, or might buffer these organisms for some of the negative consequences of environmental changes such as climate change. I address all of these using reptiles as model systems. And I do this for a number of reasons. First, I think reptiles are cool, they're fun. I always have been chasing lizards or dreaming of dinosaurs. But I actually have scientific reasons for wanting to just look at reptiles largely in this light as well. And a big reason is that temperature has really strong effects on the biology of reptiles. These organisms are ectotherms. They are unable to maintain body temperatures using metabolic heat production like mammals or birds can. And unlike a lot of other ectotherms, they're terrestrial. So they're actually exposed to really dramatic temperature changes throughout their life cycles. And environmental temperature affects their metabolic rates, their physiology, how will they reproduce, how they behave, and components of their development. Because of this, they're really high risk of climate change-induced, at least, effects, and likely decline. And even without climate change, these organisms are really highly threatened in the last year or so. There have been a plethora of work trying to get the word out that reptiles as a whole are extremely threatened taxa, They're now considered potentially the most threatened group of vertebrates. They're kind of tied with amphibians at the moment. It kind of depends on who you're talking to and whether they prefer to chase after frogs or lizards on what they tell you. But this paper just came out about 2 months ago in Biological Conservation. It's got upwards of 85 authors all trying to get behind this one statement that these organisms are in trouble and we need to understand what's going on with them and how they're going to be affected by the large amount of impending environmental variation that they're going to be exposed to. So, starting at the top, I want to address whether or not life history stages might be more sensitive to environmental variation, one life history stage versus another. And I'm going to address this question and the second question primarily using this study system. These are alligator lizards. They're native to the Pacific coast of North America. They're pretty charismatic, gregarious, feisty lizards. From the tip of their snout to the base of their tail can be about that long, and then their tail can be about twice as long, really long tails. And I work on 2 species in the same genus. They're about 6.5 million years divergent. The northern alligator lizard, Elgaria cerulea, and the southern alligator lizard, Elgaria multicarinata. These organisms are very ecologically similar. They're the same sort of size. They live in basically every habitat within their ranges. They eat anything that will fit in their mouth, and they'll try to eat things that won't fit in their mouth. These are really feisty animals. They're little dragons. But they live in different areas, even though they're basically ecologically similar. The southern alligator lizard ranges from Baja north up the coast into Washington State, whereas the northern alligator lizard ranges from the Bay Area of California north up into Canada, one of the 3 lizard species that live in Canada. And even where the ranges appear to overlap, the northern species exist at higher elevations than does the southern species. So these are basically, their geographic ranges are separated by temperature, with one species being in cold areas and the other species being in warmer areas. And I don't want to give you the impression that this is a fully static range. There's always movement in a biogeographic range, even though the books often don't change all that rapidly. And in particular, there's a lot of things happening right down here, at the southern end of the northern alligator lizard range. At least there's a lot of anecdotal evidence, none of it's been published yet, but that the northern populations of the northern alligator lizard, that just used to have tons of individuals, and I didn't mean to push that, but it's okay, are going extinct and being displaced by the southern alligator lizard. So there's some competitive interactions going on. There's some changes, potentially due to climate change, because this part of the world is warming faster than almost any other, certainly more than any other spot in the United States. And so you might expect, because they're active at different temperatures, or they live in different temperatures at least, that they would be active at different body temperatures. However, that's not the case. Very independent studies in different decades looked at the functional body temperatures of these organisms, and they're both active with mean body temperatures of about 24 degrees C, and really weird for a reptile. They could be active with body temperatures as low as 4 degrees C, so just above freezing, to 34, 35 degrees C, which is up near, that's in the 90s, up near 100 Fahrenheit. So a few degrees difference in mean temperature isn't going to be, at least on the adults and like their energetics, doesn't make sense as something that's affecting where these organisms are living. They're active at the same body temperatures. And while they have this really broad active range of temperatures that they can be active at, they do thermoregulate to some degree, at least when opportunity arises in a laboratory. The southern alligator lizard will maintain body temperatures of 28 pretty tightly when it's easy for them to. And their physiology, at least some of their enzymatic activity, is optimized at this temperature. It's hypothesized that the reason they're active in such broad ranges is so that they can use sort of suboptimal habitats, really dense scrub and be under things most of the time, where they wouldn't really have the opportunity to thermoregulate anyways. And those sorts of habitats are basically off limits to other lizards. And if you're familiar with this part of the world in particular, there are lots of lizards that they're competing with. So having access to niches that are open to them would be beneficial. So if this isn't explaining the sort of mean temperature effects on the adults, isn't really explaining their biogeography, what might be? Well, if we look a little closer, one of the first things that you'll notice about these organisms, if you look into the literature, is that the northern alligator lizard is viviparous, so they're live-bearing, whereas the southern alligator lizard is oviparous, so they're egg-laying. And we know that the gravid, or the pregnant females of the northern species, maintain really tightly body temperatures around 25 degrees, suggesting this is at least okay for development, if not optimal and preferred for development in that species. We don't yet know what temperatures are okay for development in the southern alligator lizard, but I hypothesize that differences in the thermal sensitivity of this life history stage, the embryonic life history stage, might be contributing to the biogeographic differences in the landscape we observe in these species. So then I predict that the southern alligator lizards would not be able to develop at temperatures, they wouldn't tolerate temperatures that the northern alligator lizard could tolerate. To get at this, I described the developmental thermal reaction norm for development in this species. So I went out in the field to do this, and I collected as many rabbit females as I could find by active searching, sort of flipping over rocks and logs and jumping on them, trying to avoid jumping on rattlesnakes. And they kind of look similar. They live in the same places. They both have big fat heads. It's good to have a search image. Then brought the females back to the laboratory, housed them individually in conditions suitable for oviposition, allowed them to oviposit, then exposed their eggs to different temperature treatments randomly. In the first year of the study, they were exposed to 26, 28, and 30. And then in the second year, they were exposed to those temperatures and 2 more temperatures, 24 and 32. Allowed them to incubate, then look at survivorship and the morphology of the offspring. For morphology, I was estimating things like snout bent length, which is how long they are without their tail, tail length, and body mass. And these data show the basic results for survivorship and temperature in the species. Incubation temperatures on the x-axis, percent survivorships on the y-axis, and then if they survived, it's the lighter color, and if they died, the darker color. And this is at 2 different time points. In both cases, temperature had a significant effect. But if we just look from oviposition, which is egg laying, to hatching, it's sort of a cooler is better type of reaction norm or trajectory. Those incubated at 28 or cooler had relatively high hatching success. Then it dropped off rapidly with exposure to 32 degrees C having 0 hatching success, suggesting that's around the critical thermal maximum temperature for this organism's physiology. However, if we look at a broader period, this is still before maturity, so they're going to be affecting the actual fitness of these organisms similarly. If we look from oviposition to 30 days post-hatching, actually get a more typical thermal reaction norm. It's got an optimum temperature around 28, and then the organisms do less well at cooler and warmer temperatures, at least during development. These figures are then looking to see whether there's any consistency in that sort of pattern with other phenotypes. So this is aspects of morphology. Incubation temperature is on the x-axis, and then these are 3 different aspects of morphology: snout bent length, tail length, and mass. And at the moment, I'm just showing results for at hatching. Incubation temperature did have a significant effect on all of these morphologies as a whole, using a multivariate analysis of variance. And this appears to have been driven by effects of temperature on snout bent length and tail length, both of which show a similar sort of hump-shaped reaction norm with an optimum around 27 or 28 degrees. The trend was basically consistent through 7 days of age. It's no longer significant at snout bent length, but it is for tail length and mass now, with a similar sort of hump-shaped pattern. By 30 days of age, it's breaking down a little bit more, still a significant effect on tail length. Mass still has a significant effect, but it's sort of in a different direction now. And this is probably, from what I can tell from the data, a sampling artifact of sorts, because those individuals that were incubated at 30 degrees were less likely to survive 30 days of age, and those individuals that were a little larger were more likely to survive within that group. So we ended up sort of culling out the smaller individuals, affecting this mean. Together, all these data on morphology and survivorship allow us to start to understand and describe how temperature affects the development of these organisms and describe the reaction norm. 24 degrees C induced high mortality. It's getting down near the critical thermal minimum temperature. Around 31 is going to be the critical thermal maximum. There was no survivorship at 32. And then there appears to be an optimum for development around 28. And this was held across life history traits. The morphological traits showed this sort of optimum, and survivorship showed an optimum around 28. And if you remember, this was actually the optimum for the adults as well. They had higher enzymatic function, and this is the temperature they selected when allowed to. And so it suggests that this optimum is conserved across phenotypes and life histories of this organism. However, the shape of this reaction norm is quite a bit different in the embryos than it was for the adults. It's quite narrow. I just sort of filled in what the bottom probably looks like, but I don't have data for it. These tend to curve out sort of exponentially along the tail. But generally, the effective temperature is really narrow. These individuals have poor performance when the eggs are exposed to temperatures just a few degrees below or above 28, whereas the adults, if you remember, they could be active from 4 to 35 degrees C. This suggests that there is a difference in the thermal sensitivity of the different life history stages, with the embryos being far more sensitive to temperature than are the adults. This is likely sort of an ancestral condition in the snakes and lizards, the squamate reptiles. Most squamates have this sort of narrow thermal reaction norm. And it likely is having major influences on the biology and just sort of the evolutionary history of these organisms. Because if you think about it, a rabid female lizard could be active all over the map. They can be active at lots of temperatures. But if they lay their eggs in the wrong area, they're going to die. So there's going to be really strong selection on nest site choice in these organisms. Moreover, the differences I saw reported in this reaction norm and in the different sensitivities of these lizards and the northern alligator lizard does support the hypothesis that this is affecting or contributing to the biogeographic differences in these organisms. Southern alligator lizards have poor performance and poor survivorship when they are incubated at temperatures that are really suitable for the northern alligator lizard. 25 is good for the northern alligator lizard, but that's going to be close to the critical thermal minimum temperature for the southern alligator lizard. And this might really keep the southern alligator lizard from invading the higher elevations and higher latitudes that the northern alligator lizard can persist at quite easily. However, while I do think this is contributing to their biogeography, I don't want to give the impression that this is the whole story. There could be a lot of other things involved. For example, thus far, I've really only talked about the effects of the mean environmental temperature, the average, on the biology of the adults and the life history and the embryos. But a lot more is going to happen with climate change. In particular, there can be effects of extreme temperatures. We think heat waves are going to become more common, storms and fires are going to become more common. So if these organisms differ in how they respond to extreme events, it also could affect their standing biogeography and how they're impacted by climate change. Which brings us to the second vignette that I'm going to talk about and try to address. When is temperature potentially physiologically stressful in these organisms? Because as they're exposed to more and more extreme temperatures, it could start to have a stress effect that reduces the fitness of these organisms. And I addressed this by examining corticosterone in these animals. This is a glucocorticoid hormone. Corticosterone is the reptilian stress hormone, although you'll often hear it termed the bird stress hormone. It's the stress hormone for both the avian and non-avian reptiles, I assure you. And its corticosterone responses are generally correlated with what's sort of colloquially known as the fight or flight response. And this is a generally adaptive response. These hormones are always in the plasma at relatively low levels, and they'll sort of circulate with time and go up and down. And at these levels, they just are affecting basic homeostatic function, the body sort of taking care of itself. However, when an individual is exposed to a potentially threatening situation and they become stressed, these cort levels shoot up and they spike. And this induces a suite of physiological responses, things like shutting down digestion, shutting down reproduction, it's making the organisms twitchy. It's that fight or flight response. And that can, which is generally adaptive when the individuals are exposed to a threatening experience, like a predator sneaking up on them. However, if these levels are elevated really frequently or for long periods of time, they can reduce the fitness and general survivorship of the organisms. So you can imagine if we have sort of directional climate change or any directional environmental effect, whether that's habitat destruction or pollution or the introduction of an invasive, if it elevates the stress levels of the organisms, then it could be reducing the fitness and the ability of those organisms to persist on the landscape. And while we know temperature is really important for the biology of reptiles, and we know that these stress effects can happen and high levels of stress can reduce the fitness of organisms, we don't actually know whether extreme temperatures are stressful in reptiles, even though that's a really intuitive thing to think about. It's only been examined in 3 species that I'm aware of to date, and the results were equivocal. Some species showed an effect of temperature on stress, others, there was no effect of temperature on stress reported. And these were generally field experiments. They were basically correlating daytime temperature with levels of corticosterone. And so I wanted to look at this in a more experimental framework with my co-author, Dr. Liz Addis. And what we hypothesized was similar to the differences in tolerance and life history stages, is that these differences in thermal stress could be affecting the fundamental niche of these organisms, and it could be doing so differentially and thus affecting the landscape level biogeography of these two species, the northern and southern alligators. And if this is the case, we predict that, 1, extreme temperatures will induce elevated glucocorticoid levels, but it will do so differentially, with one species, the northern species, having high levels of these hormones, being stressed at warm temperatures, the other species having high levels of these hormones and being stressed at cold temperatures. And to get at this, I used laboratory temperature manipulations of the adults of both species to try to understand what was going on with these hormones. And that's on that slide. So I again went in the field, I collected animals. This time I collected adults only, both males and females, 15 individuals of both species, and they were collected from the same basic area here in the Bay Area of California. If you'll remember, this is that area of the range that appears to be in flux, with the northern alligator lizard becoming displaced, especially their lower elevation sites becoming displaced by the southern alligator lizard. I brought them back to the laboratory and housed them individually, pretty much like I did the females from the previous experiment. And they were left in the laboratory for a year or more and allowed to have this really long-term acclimation to laboratory conditions. And then I took a baseline blood measurement to sort of get a laboratory baseline for what their port looks like. We don't know for sure if they were stressed, but we have no indication that they were stressed in the laboratory. These were all in very good body condition. They've been eating well, they've been copulating, they've been doing things lizards do. So it seems like they weren't stressed, but they might have been. And then I let them rest for 2 weeks, and then exposed them to one of 4 different temperature treatments in random order. Either a 10°C treatment, which is cold, but within the range where they're frequently active, 20, which is near their average functional body temperature, 28, which is at least the optimum temperature for the adults of the southern alligator lizard and the developing embryos of the southern alligator lizard, and then 35, which is within their active range, but it's really warm, and much hotter is fatal for this species. I then left them in these treatments for 5 hours, after which I drew blood from the animals. By the way, they were in these treatments in their home boxes. I wasn't even taking them out and putting them in another box, which could potentially be stressful. They stayed in their nice home enclosure that smelled like them in these large cabinet incubators that were lit on a normal dial cycle. After the 5 hours of me taking blood, I put them back on their normal home shelves, let them rest a week, and then did the experiment again, fully randomizing their order and putting them in. And I did this 4 times, so that every individual was exposed to every temperature treatment in random order, giving me a complete repeated measures randomized statistical design. After which, I took all the blood, spun it down into good plasma, and used a commercially available radioimmunoassay to actually quantify the corticosterone levels in the plasma. And those results are shown with these figures, these box plots. Temperature treatment, again, is on the x-axis, and change in corticosterone is on the y-axis. What these values are is for every individual for each treatment. I took their treatment value and subtracted off that baseline value, which would sort of be their standard port level, to figure out how different the treatments, how much the treatments affected their port levels from baseline. So if there was no change, we'd expect it to be right on this 0 line, and then could be above or below. There was no effect of temperature treatment, any of these temperatures, on the corticosterone level in the southern alligator lizard. However, the northern alligator lizard showed a significant effect of temperature at the 2 warmer temperatures on corticosterone. Not only were these treatments induced elevated levels that were different from the other 2 treatments, but these levels are significantly different from that 0 point, suggesting that these really are stressed organisms in the warmer, when they're exposed to the warmer treatments, but not when they're exposed to the colder treatments. In addition, it might not be immediately obvious, but the southern alligator lizard had a lot more variation in their corticosterone response to temperature. And that becomes more obvious when I plot these on the same scale. You can see there's a much broader variation in the southern alligator lizard. And if this is at all heritable, it would suggest that the southern alligator lizard has a lot more potential for evolution in their corticosterone response than does the northern alligator lizard. And these results suggest, first off, that how especially a reptile, responds to temperature as far as stress is highly species specific. These organisms are congeners. They're only about 6.5 million years divergent, but they have radically different stress responses to temperature. That could partially explain why the 3 previous studies were equivocal, with some showing a response and others not showing a response. It's just species specific. In addition, this is likely affecting the fundamental niche of these organisms and how they vary and whether or not they're becoming stressed or not at temperature might affect where they do and do not persist. And the differences we observed in the stress response with temperature is in the same direction as our predictions, or as my predictions. It suggests that the northern alligator lizard might actually, its range might be constrained to colder areas because of the stress response. When they're exposed to temperatures like 28 or 35, which really aren't that hot, that's in the 80s and 90s, which you can believe happens down here all the time, they're going to be highly stressed, whereas the southern alligator lizard is not. And even if these stress levels aren't enough to be actively killing individuals, if it makes them hide, or not be reproducing, or not digesting, then it's going to make them competitively inferior to the southern alligator lizard. It's going to completely change the competitive landscape and allow this species to outcompete this species. And that might be a little bit of what's going on down here, though obviously we need a lot more work on the ground to figure out whether that hypothesis is valid. So now I've talked quite a bit about these alligator lizards, and I'm going to change gears a bit. And I'm going to address this sort of behavioral plasticity question, whether or not timing of reproduction might buffer organisms from climate change. And I'm going to change away from the alligator lizards and talk about painted turtles for a bit. These are the primary study organism of the laboratory I'm in, Fred Jansen's lab. He's been working with these organisms for over 25 years now, primarily at a single nesting location, which is an island on the Mississippi River. It's on the Illinois side, bordering Illinois and Iowa. And this island has really a very small area. It's about the size of this building footprint-wise. It's one of the largest known nesting beaches of any freshwater turtle. We get about 300 or more nests a year of this species, all in this little area. It's actually an RV park. It's kind of funny. People don't even notice that there are these turtles digging nests everywhere. And it's allowed us to have a lot of physiological data on these organisms, as well as just field nest temperatures. And these are the types of data that are necessary for building a mechanistic model. So what we did to address this question was actually build a mechanistic model, though it's not spatially explicit, it's more temporally explicit, to try to understand whether or not how reproductive timing and climate change might interact to affect developing embryos. And the reason why I think this is a really timely thing to be addressing is that the most commonly observed biotic response to climate change are changes in the timing of reproduction and other life history events. These are called phenological responses. We see flowers blooming earlier in the year, we see tree buds bursting earlier, butterflies and birds are migrating earlier in the year, frogs and crickets and all sorts of things are singing earlier in the year, and things like lizards and birds and lots of organisms are nesting and reproducing earlier in the year in response to warming temperatures. And it's hypothesized that this might allow organisms to sort of match their life history events with suitable temperatures. Because as it gets warmer, if you nest earlier in the spring when it's still cooler, then you might be able to nest at the right temperature. And this could be really important in a number of reptiles. I showed earlier with my very first little vignette that temperature can affect the development of reptiles quite a bit. Things like body shape and size. But one thing that alligator lizards don't do, but a lot of reptiles do, is temperature can define the sex of these organisms. And it's just temperature during a short period during development. It's called the thermosensitive period. It's the middle third of development. It's often about a month of time. And that 1 month, and the temperature the embryo experiences during that 1 month, affects the most important demographic parameter that organism is ever going to know, whether or not it is male or female. And really subtle differences in temperature can affect differences of just 1 or 2 degrees. And this figure shows the most common pattern of temperature-dependent sex determination, or TSD. This is called type 1 TSD. It's colloquially referred to as the hot chicks and cool dudes model, because we have the chicks being developed at hot temperatures and the dudes being developed at cold temperatures. It's a very crafty way of doing this. But you get the basic idea. And there's a range in the middle that I had shaded, but I don't have that showing up, where you can get mixed sexes. And this range is generally only 1 or 2 degrees wide, sometimes as many as 4, but that's about as broad as it is. So if you change temperatures that embryos are experiencing by just a few degrees, you can go from being completely male to completely female, or from mixed to one or the other. And the temperature in the middle of this curve is called the pivotal temperature. That's the only point where you expect to get a 50-50 sex ratio, a pure 50-50 sex ratio. And that's the sex ratio that most mammals and birds, and it's most common in vertebrates, we get through segregation of sex chromosomes, genotypic sex determination. These don't have any sex chromosomes. So because this one parameter, sex, is so dependent on temperature, these organisms are thought to be potentially at really high risk of climate change-induced decline. Because directional climate change in particular, that thermometer just marching up 4 to 8 degrees, has the potential to shift you completely out of this zone and into this zone, where you only have a single sex. You then reduce effective population sizes and increase the probability of population extinction. However, it's been proposed on numerous occasions that maybe females by nesting earlier will be able to buffer their progeny from these effects. This is a really attractive hypothesis because we know from a lot of fieldwork that lots of species with temperature and sex determination are nesting earlier in the year in response to climate change. So we wanted to test this question, and we realized pretty quickly that it's going to, fundamentally, whether or not this works will depend on how well females can predict temperatures during that thermosensitive period when they actually lay their nests. And this might be kind of tricky because the thermosensitive period, at least in turtles and lots of other organisms, beginning the middle third of development, is as much as a month after the females actually put their eggs in the ground. And whether or not the temperature when they put their eggs in the ground is predicted will depend on sort of seasonal patterns of temperature change. To describe that a little better, I have this cartoon. Seasons on the x-axis, temperatures on the y-axis. It gets warmer in spring, levels off in midsummer, and then gets colder in autumn. The next year you'd see just sort of swing back up. Kind of looks like a sine wave. And if we have females nesting in the thermosensitive period before climate change, and we say this might be June 1st, which is the average nesting date historically of our painted turtles at our site on the Mississippi River. And in the thermosensitive period, the time when temperatures affecting sex is roughly July, those 30 days. If temperatures then get uniformly warmer across the seasons, the females adjust their nesting date, such that they're nesting at the exact same temperature as they were before climate change. The thermosensitive period isn't falling at the exact same temperature. We've got nesting date at the same temperature. Even the onset of the thermosensitive period in this cartoon is the same temperature. But the majority of the thermosensitive period is warmer than it was before climate change, simply because it sits on a different portion of this curve. And so we wanted to know whether or not this will actually be, whether or not this curve is sufficient to cause problems, and whether or not females might be able to nest earlier in the year and actually buffer their progeny from the effects of climate change. And if that works, what we would predict is that by nesting earlier in the year at the exact same temperature every year, we'd expect that nest temperatures during the thermosensitive period will not change, and sex ratios won't change. They'll stay mixed like they historically have been in this population. So to try to get at this, I changed gears a bit. I didn't use a nice sort of classic experiment putting individuals at different temperatures, because that would be very logistically problematic. You'd have to take whole turtles that are long-lived, or basically populations of them, and put them in like aircraft hangar-sized ponds that you control the temperature of, and then somehow manipulate exactly their nesting date without affecting other things. So it would be a nightmare. But we can use mathematical models to get at this. And what this allows us to do is use what we know about these organisms and simulate predicted changes in climate and see how that will affect the sex ratio of the animals after climate change and various degrees of climate change. And so the main model that we use is a modified version of Arthur George's constant temperature equivalent model. And the reason this model was developed was to be able to translate data from our laboratory experiments and use them in the field. So laboratory incubation experiments generally are done at a single temperature. You put egg in box, leave it at a temperature until it hatches, see what happens. But in the field, in a real nest, temperatures don't stay constant. They're constant, they go up and down every day with the afternoon high and the nighttime low. So what this model was originally designed to do is allow you to crunch all of that variation from daily temperature variation down into a single number that you can use to predict sex ratio. And the way it works is if you take those sort of temperature data from a nest, calculate a CTE value of, say, 25 degrees, and you predict that nest to be the same sex ratio as if you'd taken all those eggs and put them in a box, an incubator, at 25 degrees C for the entirety of development. So, what the original model does is it just uses a cosine wave, simulate daily temperature going up and down. What we did to include seasonal temperature change was add a slope term to this model. We did a few other things, but that's the main thing. What this allows us to do is use real data, instead of this cartoon, but I'm going to show the cartoon, for how temperature changes with the environment, and we can then fit this temperature change to each one of those slopes, every slope in between. We're going to have it starting at different temperatures, do all sorts of variants with how we predict that climate change might happen We can then calculate the CTE and thus predict sex ratios given different levels of climate change and different levels of when the individuals are nesting in the season. Again, we did this work using data on the painted turtles. This was really useful because we have so much data on these organisms. We were able to estimate the physiological parameters such as the pivotal temperature and the range where you can have mixed sexes, and when it's too hot for them to develop, when it's too cold for them to develop, based on the long-term research Fred's been doing, my advisor. And then we also have lots of local temperature data that we could apply to this, and climate change model predictions that we can apply to this question. And I'm going to show the results on this figure. Can you see a shaded range here? Is it visible? Okay. Well, that's unfortunate. The x-axis is temperature increase with climate change, and that's the full magnitude of temperatures that are really predicted in the Midwestern United States over the 21st century. You might have 0 change, but that's unlikely. All the way up to potentially 6, and most models are predicting around 4 degrees C, which is on the neighborhood of 8 degrees Fahrenheit, increase in temperature over the 21st century on average in the Midwestern United States. Then constant temperature equivalent is on the y-axis. Again, this number really is just to understand what the sex ratio is going to be. So I divided up the figure based on the sex ratio that we predict. If the temperature is in this shaded range, should be a mixed sex ratio we predict from that nest. If it happens to fall right on this green line, that's the pivotal temperature where we'd expect to have a 50-50 sex ratio in painted turtles. If it's above that shaded range, we expect to have all female individuals. If it's below that shaded range, all male individuals. So to first start understanding what this model predicts, I want to look at how the sex ratio is affected, or predicted to be affected, if females don't adjust their nesting date. So if it's warmer and they just nest at the same point and the thermosensitive period is at the same point in time. This line predicts how the CTE values given different levels of climate change. And with as little as about ½ degree C, quite a bit less than 1 degree C, we expect to transition from mixed-sex nests to complete 100% female nests, which, if you actually go that far, it's population extinction. There's a lot of sort of swerviness going on in this line, and the reason for that is that this peak indicates the point when nest temperatures start getting above the developmental maximum temperature in painted turtles. So development actually stops above this temperature. While this really short-term exposure to those sorts of temperatures is not fatal or damaging, longer-term exposure becomes damaging, and extended exposure to temperatures above this critical thermal maximum is fatal. So then what do we expect to happen if females do adjust the nest temperature? So for this next line, I'm going to assume that females are able to nest at the exact same temperature every year, the same temperature they've been nesting at historically at our population. And this actually is being kind of kind to these turtles' abilities. If you actually had a 4 degrees C change in temperature, that means they'd be nesting 3 weeks early. A 6 degree temperature means they'd be nesting something like 5 weeks earlier than they are right now, which might not be possible in these organisms. We're going to see whether or not what would happen if they could do that. And what we predict to happen if this would be a successful buffering mechanism is we predict basically a straight line, or even if it curves up, to stay within this shaded range where mixed sexes occur. However, the predictions from the model are this line. We still have to predict sex ratios to become completely female with less than a degree C increase in temperature. And we still see this peaking, this plateau, which is where nests are spending large amounts of time above their critical thermal maximum, suggesting we might not even have nest survivorship, much less have completely female biased sex ratios. And this happens really at fairly low temperatures. This is just a 2 degree C increase in temperature, which really no one is confident will happen in the next 100 years. We're really expecting to see much more climate change than this. So what this model suggests is changing phenology alone is not enough to buffer these populations from the negative consequences of climate change. We expect 100% female individuals in these populations, and potentially hatchlings to not even emerge, just nest death, which would obviously be bad for these populations. And these results are likely transferable because the only thing we really manipulated in our model was when individuals were nesting. We just took into account how temperatures change with the seasons. It's really any system that is temperature sensitive and is in a temperate zone should have similar results, where phenology really isn't going to be buffering those organisms from the negative consequences of climate change. That isn't to say phenology and advancing nesting age is never adaptive or not adaptive. It could allow individuals to have longer growing seasons, or get out and be reproducing earlier in the year, and thus reducing the risks of dying before they have the ability to reproduce. But it suggests that this isn't somehow some adaptation, sort of species-level adaptation to dealing with the occasional occurrences of massive climate changes. And there are other ways these organisms might be able to buffer themselves from climate change, things like digging deeper nests or whatnot. But we really aren't seeing the kind of changes in plastic traits that lead us to believe that we're not going to see these sort of dramatic effects on the population. So this immensely large population of nesting painted turtles in the Mississippi seems to be potentially in dire straits, and that could be the case for lots and lots of organisms. So, in summary, I tried to describe sort of how important the physiology, understanding the physiology of organisms are for us predicting how they will respond to climate change. We can't just use basic correlations where we think they might be. We need to understand what sets their critical limits. And what life history phases are important. If you just model adult alligator lizards, potentially adults of any lizard, you might not be getting the whole story, especially if the adults aren't the most sensitive. In the alligator lizards, the embryos have much greater sensitivity to temperature than did the adults, and it seems to be contributing potentially to the landscape level biogeography of this genus. The southern alligator lizard cannot develop well at temperatures that are common or optimum for the northern alligator lizard, and that might keep them from being able to invade these colder climates. In addition, temperature extremes might be important not only for how these organisms respond to climate change, but just setting landscape level biogeography. We have, in the same set of species, differences in how they respond, how their stress response works with temperature. Again, this could be affecting their landscape level biogeography. The northern alligator lizard shows signs of stress at warm temperatures that do not stress the southern alligator lizard. And that might keep them from being able to evade down, really be able to live in warmer areas. And it also means that as climate changes, especially down where they're just mountain species, as it gets warmer and warmer, they're going to be sort of metaphorically chased off the mountaintops. As the southern alligator lizard is able to invade more and more and more, it'll just work right up until there's nowhere higher to go. And then we try to look at whether or not this really common biotic response to climate change, nesting earlier in the year, might really help buffer progeny from the negative consequences of climate change. And it appears that, at least in our painted turtle system, no, this isn't going to function that way. And that's largely because of how it gets warmer in the spring, faster. You've got this accelerated warming event. And so I argue that all of these things need to be taken into account when we're trying to understand how climate change will affect organisms. And really, reptiles and lots of organisms are very sensitive to the abiotic environment. And the changes that are happening right now really do threaten these organisms. And without some action and some more understanding, we might not have things like painted turtles in 100 years. That, I'd like to thank everyone that helped me. I had lots of help. Co-authors, Dr. Liz Addis, Fred Jansen, and, who am I forgetting? Oh, Karen Abbott, math whiz. Lots of people helped, either with comments or assistance in the field, various agencies like the U.S. Army Corps, the SLV Water Department, and the BLM for allowing me access to collection sites, various funding agencies, and thank you all very much for allowing me to come back to UCO and tell you about what I've been up to. Thank you.